Alanine racemase and its potential for antimicrobial therapy. Section 1. Overview of racemases.
In this video, we will be discussing alanine racemase. This enzyme belongs to the family of isomerases, which rearranges a molecule structure. Racemase, in particular, alters the chirality of a given molecule, in this case, alanine.
Alanine racemase is naturally found in bacteria which use this enzyme to construct their peptidoglycan layer. The peptidoglycan layer is a mesh of polymers right outside the plasma membrane, consisting of saccharide subunits and amino acids. It forms the backbone of bacterial cell walls. This layer contributes to the structural stability of the bacteria and plays an important role in the bacterial cell reproduction process of binary fission.
Thus, anything that would disrupt the catalytic activity of alanine racemase could serve to weaken the bacteria's cell wall. This attack on the cell wall is similar to the effect of penicillin on bacteria. Let's take a look at the structure of alanine racemase.
Section 2. Structure of alanine racemase.
Crystallographic studies by Xiao et al. Of alanine racemase have revealed its three-dimensional structure. Alanine racemase has two domains, one dominated by alpha helices surrounded by beta sheets and the active site, and the other dominated by beta sheets and including the N-terminus.
Now, let's take a look at the proposed mechanism of alanine racemization.
Section 3. Mechanism of catalysis. First, it is important to note the cofactor at the center of this proposed mechanism, pyridoxal 5' phosphate, also known as PLP.
Here we see PLP with the formal group replaced by the R group of a lysine. Usually the nitrogen in the pyridine ring of PLP is protonated. This is due to the high pKa usually associated with nitrogen atoms bearing a lone pair of electrons compared to the relatively neutral environment in which they exist. However, quantum mechanical and molecular dynamical simulations by Rubinstein and Major in 2010 have shown that the proximity of arginine 219 contributes greatly to the stabilization of the unprotonated species.
The bond between lysine and PLP is first displaced by the amino group of L-alanine, attaching the alanine to the cofactor. Notice that the alanine is stabilized by hydrogen bonding from a nearby tyrosine. Then, the tyrosine exchanges protons with the alanine, deprotonating the alpha carbon while protonating the carboxylic acid. This intermediate is only possible due to the heavy stabilization offered by hydrogen bonds.
Finally, the protonated R group of the lysine protonates the alpha carbon from the opposite side of the original hydrogen, reversing the alanine's chirality when the lysine displaces it. Thus, L-alanine is converted into D-alanine, and the PLP-lysine complex is reformed. Section 4. Research into racemases.
As we previously mentioned, D-alanine is a key building block of prokaryotic cell walls. Because alanine racemases are absent in eukaryotes, but ubiquitous among prokaryotes, this enzyme is an attractive antimicrobial target.
A recent study conducted by Liu et al. In 2015 focused on alanine racemase in Aeromonas hydrophila, which causes a chronic infectious disease of freshwater fish. The study investigated the alanine racemase gene AIR2. Plasmids were designed with the knockout AIR2 gene and transduced into Aeromonas hydrophila, thereby disrupting the production of alanine racemase in the bacteria.
Both wild-type and mutant groups were subjected to D-alanine starvation, eliminating environmental factors, and the transmission electron micrographs of the strains are shown side by side at 0, 20, 30, and 40 hours. At 0 hours, the wild-type and mutant cells showed relatively the same morphology.
Defects in the cell envelope began to appear after 20 hours, and the cells displayed continuing damage in their wall integrity as time increased. Many mutant cell membranes had holes, as indicated by the arrows, and the cell substances leaked out of the cell.
Observation of these bacterial strains also seemed to indicate that the cell wall damage of bacteria mainly start in the polar regions, a result consistent with previous findings that support the preferential interaction of antimicrobial peptides with negatively charged polar regions of E. coli cells. This is a promising area of further study. All in all, these studies show that alanine racemase is a legitimate target for potential antibacterials.
While the exact mechanism of the interconversion of D-alanine and L-alanine by alanine racemases has not been fully delineated, the potential medical benefits of disrupting alanine racemase activity include novel methods of treating diseases caused by bacteria.